Welcome to channel 360. Today our topic is Postmortem changes, which is timeline of death. How we determine the time and type of death, and see how it was occurred. But before we proceed to our video, there is a small request that if you're new to channel, please like and subscribe to channel so that you get notified about every incoming videos. Thank you very much. Let's start our new video. Postmortem changes, unraveling the timeline of death. This presentation delves into the fascinating world of postmortem changes, exploring the scientific principles behind how a body transforms after death. We'll unravel the complexities of estimating time of death, a critical aspect of forensic investigations. Understanding the forensic science of postmortem interval. Postmortem interval (PMI), the time elapsed between death and the discovery of the body, also known as the PMI, is a crucial factor in forensic investigations. It provides vital insights into the events surrounding the death. The postmortem interval PMI is the time between death and when a body is found. Think of it like a clock that starts ticking at death. Forensic experts use clues from the body like temperature or decomposition to estimate this time. Knowing PMI helps piece together what happened, like when or how someone died. It's a key puzzle piece in solving mysteries, turning time into evidence. Forensic science. Forensic scientists use a variety of techniques to estimate the PMI, relying on a combination of physical and chemical changes that occur in the body after death. These changes are predictable and offer valuable clues. Forensic experts estimate PMI by tracking predictable physical and chemical changes in the body after death. Think of it like a stopped clock. Temperature drops, tissues decompose, and chemicals shift. These time processes act as clues. By analyzing them, scientists pin down the time since death, reconstructing case timelines. It's nature's clock, decoded by science. Algor Mortis, the cooling signature of death. Cooling rate. The body loses heat after death at a predictable rate, a phenomenon known as Algor Mortis. This rate is influenced by factors like air temperature, clothing, and body size. Algor Mortis is the cooling of the body after death, like a hot drink left out. After the heart stops, the body loses heat at a predictable rate, but this cooling clock is influenced by the environment like air temperature, body size, clothing, and even humidity. Forensic scientists measure body temperature and compare it to these factors to estimate time since death. For example, a large body in warm clothes cools slower than a small one in cold air. While not perfect, Algor Mortis helps narrow down the postmortem interval PMI, acting as a key clue in reconstructing timelines during investigations. It's one piece of the puzzle, combined with other changes like rigor mortis or decomposition, to solve the mystery of when in forensic cases. Temperature measurement. Forensic scientists use rectal temperature measurements to estimate the PMI, as the core body temperature changes more slowly than the surface. Forensic scientists measure rectal temperature to estimate time since death because the body's core cools slower than the skin, like how a baked potato's center stays hot longer than its skin. They insert a thermometer into the rectum to track this gradual heat loss, then use formulas that factor in air temperature, body size, and surroundings. While helpful, this method isn't perfect. Extreme weather or clothing can skew results. It's one tool in a detective's toolkit, paired with other clues like rigor mortis to build a clearer timeline of death. Limitations. Algor Mortis is not a perfect indicator of time of death. It can be affected by environmental factors, and the cooling rate slows down as the body approaches ambient temperature. Algor Mortis body cooling helps estimate time of death, but it's not foolproof. Think of it like guessing how long a pizza's been out. Cold air cools it faster, but a blanket clothing slows cooling. Once the pizza matches room temperature, you can't tell how long it's been cold. Similarly, once a body reaches ambient temperature, the cooling clock stalls. Weather, body size, or sudden temperature shifts, like a body moved indoors, also skew results. That's why forensic experts combine Algor Mortis with rigor mortis, decomposition, and insect activity. No single clue tells the full story. Rigor mortis. Muscular stiffness and its investigative significance. Muscle stiffness. Rigor mortis is the stiffening of the muscles after death. The muscles start to stiffen within a few hours, reaching peak stiffness around 12 hours, and then gradually relaxing. Rigor mortis is the stiffening of muscles after death, like a door slowly locking. It begins 2 to 4 hours post-death as energy stores deplete, causing muscle fibers to freeze. Stiffness peaks around 12 hours, then fades over 1 to 3 days as tissues break down. Forensic experts use this timeline to estimate time of death, but it's not foolproof. Heat speeds up stiffening like a door warping in summer, cold slows it, and intense exercise before death can trigger rigor faster. Think of it as a biological stopwatch, but one that's easily nudged by external factors. To get accurate results, scientists pair rigor with body temperature, Algor Mortis, decomposition stages, and insect activity. Each clue fills gaps the others leave, creating a fuller picture of when death occurred. It's detective work, powered by biology. Time estimation, the onset, progression, and relaxation of rigor mortis can help forensic scientists estimate the time of death, although it's not an exact science. According to rigor mortis, the stiffening of muscles after death acts like a rough biological clock for forensic scientists. It starts 2 to 4 hours post-death as energy ATP runs out, locking muscles in place. Stiffness peaks around 12 hours, then fades over 1 to 3 days as tissues break down. While this timeline helps estimate time of death, it's not precise. Heat speeds up the process. For example, a body in summer stiffens faster, cold slows it, and exercise before death can trigger early rigor. Think of it as a dimmer switch, not an on-off button. To improve accuracy, experts pair it with body cooling, Algor Mortis, lividity, blood pooling, and decomposition. For example, a body found stiff and cold suggests a longer PMI than one still warm. Rigor is a clue, not a stopwatch. Context matters. Influencing factors such as temperature, physical activity before death, and body weight can influence the onset and duration of rigor mortis. Rigor mortis, the post-death muscle stiffening, is shaped by 3 key factors. Temperature acts like a chemical reactions accelerator. Heat speeds up rigor. For example, a body in a hot room stiffens faster, while cold slows it. Physical activity before death matters too. Exercise drains ATP energy, hastening rigor's onset. For example, a runner's muscles lock sooner than a resting person's. Body weight plays a role. Larger bodies retain heat longer, like insulation, delaying rigor, while leaner bodies cool faster. These variables mean rigor isn't a precise clock but a clue. Forensic experts combine it with body cooling, lividity, and decomposition to estimate time of death accurately. It's teamwork, not a solo act. Let's look to what Livor Mortis said about blood settling and post-mortem movement insights. In initial stage, Livor Mortis, or hypostasis, is the pooling of blood in the lowest parts of the body due to gravity. It starts to appear within 1 to 2 hours after death. Second is visible change. The blood pooling creates a purplish discoloration on the skin, visible within 3 to 4 hours. It helps determine the position of the body at death. Third and final is fixed stage. After 8 to 12 hours, the Livor Mortis becomes fixed, meaning the discoloration will remain even if the body's position is changed. This can indicate if the body was moved after death. Livor Mortis, or hypostasis, is like gravity's fingerprint on a body. After death, blood sinks to the lowest areas, creating purple-red patches on the skin. Think of paint pooling in a tilted bucket. This starts within 1 to 2 hours, becomes visible in 3 to 4 hours, and fixes after 8 to 12 hours. Once fixed, the discoloration stays even if the body is moved. If the patches don't match the body's final position, it suggests someone moved it post-death. For example, a body found face-up with patches on the back was likely moved hours later. It's nature's timestamp, helping detectives reconstruct crime scenes. Now look what is vitreous humor analysis, a biochemical window into time of death. 1. Is vitreous humor. The clear gel inside the eye changes its chemical composition after death, providing valuable clues for PMI estimation. Second is potassium levels in the vitreous humor rise after death at a predictable rate, allowing scientists to estimate the time since death with varying accuracy. And now third is other biochemical changes, such as the accumulation of certain proteins and sugars, can also contribute to PMI estimation. The eye's vitreous humor jelly-like fluid acts as a chemical clock after death. Like juice in a sealed cup, it's protected from quick contamination. Potassium levels here rise steadily post-mortem, giving scientists a timer. Higher potassium approximately equals longer since death. Proteins and sugars also shift predictably, adding extra clues. While not perfect, factors like age or disease can tweak results. This method shines in early PMI estimation, first 48 hours when body cooling or rigor are less reliable. Think of it as a backup clock, quietly ticking inside the eye, helping forensics piece together the win of death. Now look into environmental factors influencing post-mortem decomposition. First is temperature. Higher temperatures accelerate decomposition, while colder temperatures slow it down. This can significantly affect the accuracy of PMI estimation. Second is humidity. High humidity promotes bacterial growth, accelerating decomposition, while low humidity inhibits bacterial activity and slows down the process. Third is insects. Insects play a significant role in decomposition, attracted by the body's odor. The type and stage of insect development can help determine the time of death. Decomposition. Nature's recycling process relies heavily on temperature and humidity. Heat speeds decay like leftovers, spoiling faster in summer, while cold slows it. Humidity acts as a bacterial turbocharge, turning tissues mushy, whereas dry air preserves them. Insects, like blowflies, swarm early, laying eggs that hatch into maggots, each life stage acting as a biological timestamp. But these clues aren't foolproof. A body moved from a hot car to a fridge, or scavenged by animals, scrambles the timeline. That's why forensic teams blend decomposition clues with body cooling Algor Mortis, muscle stiffening Rigor Mortis, and blood pooling Livor Mortis. Together, they turn nature's messy process into a detective's toolkit, balancing science's limits with creative problem solving. Let's look into the challenges in accurately estimating time of death. First is individual variation. The rate of post-mortem changes can vary depending on factors such as age, health, and cause of death. Second is environmental factors. Environmental factors like temperature, humidity, and insect activity can significantly influence decomposition rates. Third is time of death uncertainty. It's not always possible to determine the exact time of death, especially if the body has been discovered long after the event. Post-mortem changes are like a biological stopwatch, but its speed depends on the body's history, age, health, and surroundings. A young, healthy body decays slower than an older or sick one. Hot, humid environments act like a fast-forward button for decomposition, while cold or dry conditions press pause. Insects. Nature's cleanup crew add timelines through delaying cycles. Yet, if a body is found weeks later, clues blur like faded ink. No single method, rigor mortis, Livor mortis, vitreous humor, gives exact answers, so forensic teams blend them, accepting gaps. It's science's best guess, balancing nature's chaos with human logic. Let's have a short look into forensic techniques and methodological considerations. Physical observations. Forensic scientists carefully examine the body for post-mortem changes such as Algor, Rigor, and Livor mortis to gain insights into the time of death. Biochemical analysis. Samples of body fluids like vitreous humor are analyzed for biochemical changes that occur after death, providing valuable data for PMI estimation. Entomological evidence. The presence and stage of insect development on the body can help determine the time of death, especially in cases where the body has been exposed to the elements. Scientific validation. Forensic techniques are constantly being refined and validated to ensure accuracy and reliability in PMI estimations. Forensic scientists act as detectives of biology, piecing together the win of death using three key bodily changes: Algor mortis body cooling, rigor mortis muscle stiffening, and Livor mortis blood pooling act like biological clocks. For example, a cold, stiff body with fixed purple patches suggests death occurred hours prior. But they dig deeper. The eye's vitreous humor, a jelly-like fluid, is tested for rising potassium levels, offering chemical clues when traditional methods fade. Meanwhile, insects like blowflies serve as nature's stopwatch. Maggot stages hint at days since death. Yet, no single method is perfect. A body in a hot swamp decomposes faster than one in a fridge, and a sick person's tissues may decay unusually. That's why modern forensics blends these tools, calibrating them with environmental data, temperature, humidity, and victim-specific factors, age, health. Labs constantly refine techniques, using controlled studies to improve accuracy. Think of it as a science-powered puzzle. Each clue narrows the window, turning decay into data and uncertainty into answers. Now move to the conclusion. Integrating multiple indicators for precise forensic assessment. Estimating the time of death is a complex process that requires a thorough understanding of post-mortem changes. By integrating multiple indicators, including Algor Mortis, Rigor Mortis, Livor Mortis, Vitreous humor analysis, and entomological evidence, forensic scientists can provide a more precise assessment of the time elapsed since death. Estimating time of death is like solving a puzzle with pieces that fit imperfectly. Algor Mortis body cooling acts as a thermometer. Imagine a coffee cup losing heat. Rigor Mortis muscle stiffening is a biological lock, tightening and loosening over hours. Livor Mortis blood pooling paints gravity's fingerprint, showing if a body was moved. Meanwhile, the eye's vitreous humor offers a chemical clock. Rising potassium levels hint at early hours post-death. Insects, like blowfly maggots, add timelines through life stages. But no single clue is flawless. Heat warps cooling rates, exercise speeds rigor, and rain washes away insect evidence, so forensic experts layer these clues. For example, a cold, stiff body with fixed lividity and fly eggs suggests death approximately 12 to 24 hours prior. Labs validate methods using real-world scenarios, for example, studying decomposition in different climates to reduce guesswork. It's science's best effort to decode death's timeline, balancing nature's chaos with meticulous observation. That's all for today. Hit like, comment below, and share this video to your friends and family. Please do subscribe if you like to watch more videos like this. Thanks for watching. Goodbye.